All right, good afternoon, and thank you for joining us for another seminar this week of midterms and week of a couple of faculty candidates. We're not meeting a faculty candidate today, we're having a guest speaker. So, and today's speaker is Dr. Blaine Mooers from the University of Oklahoma, Health Sciences Center, Department of Biochemistry and Computational Biology. I met Dr. Mooers about maybe 3 years ago. We're both on a grant writing workshop over Zoom. So we both turned out for a few weeks in a row, and then I dropped out at some point, and then I heard and I realized from Blaine afterwards that he also dropped out a few weeks later. So I don't feel like such a big dropout. It must have been not that great a workshop after all, but we actually met back there, and then we've been keeping in touch ever since. And our research interests are quite complementary. He did a PhD at University of at Oregon State University in Corvallis, and then he was a fellow with the Howard Hughes Medical Institute at University of Oregon. His work is computational biology and computational chemistry. He's going to talk to us about his work today on using imaging for RNA editing for trypanosomes. Blaine's been involved in quite a few research grants from NIH. He's also been on the study section for several NIH initiatives. He's also the two-time chair of the Stanford Synchrotron Radiation Light Source User Executive Committee, which is essentially an umbrella grant or program over 2,200 laboratories. And as I mentioned before, his research interests lie at the intersection of developing software for improving computational workflows in structural biology. He uses a lot of Python and Jupyter for a lot of software development for data analysis. And as I mentioned, he also uses X-ray crystallography, small angle X-ray image scattering, structure-based drug design, and molecular dynamics. So we will hear from Dr. Mooers today on his presentation about using X-ray photons to study RNA editing in trypanosomes and tools to ease the use of PyMOL. And I have an idea what trypanosomes are. I'm a biomedical engineer by PhD training. I've heard that word before. I'm not sure how many of you have heard that word, so we'll let Blaine explain to us what that is. Thank you so much.
Thank you very much, Christian, for that wonderful introduction. So he's covered my title. I'm going to try to follow the recommended structure for the seminar, in which I'm supposed to spend a minute or two talking about my background. So I went to undergraduate school in Missoula, Montana. I actually majored in natural resources, and I afterwards went to work for the federal government for a few years out on the what was called the Eastern Front in Montana, otherwise the North Dakota border, and I didn't really like it. So I decided to go to graduate school. I got into Oregon State University and in Corvallis, Oregon, and I had to spend a couple of years boosting up my background in math, physics, and chemistry. And I was able to get into the biochemistry and biophysics program. I worked with Xing Ho on studying the structures of unusual DNA molecules. So his lab was focused on left-handed zDNA, and I wound up crystallizing and characterizing the structures of several other unusual DNAs, and in so doing, learned quite a bit about crystallography. Up the river, the Willamette River in Western Oregon, is Eugene, Oregon, and Brian Matthews was located there. He's a leading protein crystallographer who was using crystallography to address questions about how protein sequence affects protein structure and affects its thermodynamic stability. So these questions are very similar to the questions my graduate work was focused on, and it was a sort of a natural transition to make. So after working on his primary project, I was able to move into working on the structure of RNA. I determined the first structure of a triplet repeat RNA, an RNA associated with myotonic dystrophy. I then started work on structures of RNAs associated with this very unusual RNA editing system, which I use as a job talk subject. And so I use this in my job proposal to land a faculty position at the University of Oklahoma Health Sciences Center in Oklahoma City. This is a view. You can see that my horizon has flattened or widened. There are no mountains in the background. But, and that yellow arrow points to my laboratory. I'm located in some very prime real estate for doing biomedical research. So for next 5 slides or so, I'm going to talk about this RNA editing system in trypanosomes. And I'll do my best not to lose you. Don't worry, I'm going to move on after the slides to some nuts and bolts of crystallography, which might be easier to relate to. But on the left, we have a line drawing of a single cell organism, a trypanosome. It has a flagellum that is attached to the along the edge of the cell, and it has a little end that's free. And this flagellum is used to cause the body of this protozoan to make a wavy motion, and that motion is used to propel it. Here's one trypanosome in amongst some red blood cells. They have a darkly staining body located here. You can see this it's stained with a blue dye. And initially it was thought that this was the motor that was used to drive that flagellum. But actually, further work with electron microscopy revealed that there was this darkly staining body is actually a bundle of mitochondrial DNA. And this mass of mitochondrial DNA is actually composed of 2 kinds of DNA. So we have mitochondria. We don't have one, we have dozens of them in our cells. And each mitochondria has within it a maxi-circle DNA that encodes 15 genes for proteins that are part of the electron transport chain, and 2 ribosomal genes. These ribosomal RNA genes are used in assembly of a ribosome that's used to make protein, these 15 proteins inside of the mitochondria. The mitochondria are actually composed of several hundred different proteins. Most of those proteins their genes are encoded in the nuclear DNA. But mitochondria have come up with this system of synthesizing the electron transport chain proteins inside of the mitochondria because they're so critical to the functioning of the mitochondria. Then there's a second genome composed of mini-circle DNAs. There are only 1,000 base pairs in length compared to the maxi-circle DNAs that are 20,000 base pairs in length. So this is not found in humans. And these mini-circle DNAs are vast in number and vast in kind. So there's 17, they encode 1,700 guide RNA genes. There's over 600 of these mini-circle DNAs. So there's 2 to 3 guide RNA genes per mini-circle DNA. The guide RNAs are used to edit most of the protein encoding transcripts. This is an extreme example of the editing process. This is the base sequence for a mRNA. This is the 5' end, and at the other end is the 3' end. And so we have directionality here. The sequence in black is the original sequence that was transcribed from the DNA. The U's that are in red were inserted by the editing process, and the blue dots correspond to sites where U's were deleted. And the U that I'm talking about is not just the base of the nucleotide, but the entire nucleotide, the phosphose and ribose, the phosphate and ribose are removed along with the base. And whenever U's removed, they're inserted. And this is unlike the editing that's found in humans, where usually there's just a change in the base rather than insertion or deletion of an entire nucleotide. So this editing system is not found in humans, and as a result, there's a lot of interest in this editing system for drug targeting. So these guide RNAs are run the show. They don't do the actual work of cleaving the mRNA and inserting full nucleotides. That's done by proteins. located in a big complex called the editosome. This complex has 40 proteins, and it's about the size of the ribosome. But the guide RNAs, through base pairing with the mRNA, are actually running the show. So we have a mRNA stretched out in this cartoon. On the left side, by convention, we have the 5' end. On the right side, the 3' end. NE means not edited, and this hashed area is going to be edited, but has not yet been edited. So we have a guide RNA number 1 will bind. As I mentioned, there are 1,700 different guide RNAs. They differ in their sequence, they differ in their length. So they have 3 functional domains. One is an anchor domain that binds to the mRNA by base pairing, and so uses Watson-Crick base pairing and GeoWobble base pairing. And then there's a sequence that's represented by this loop that is the template domain, and it has sequence that will complement the final edited sequence in the mRNA. So this red area corresponds to a sequence in the mRNA where we've had U's inserted or deleted as dictated by that template domain. And so after the editing process, we have base pair complementarity. We have Watson-Crick or GeoWobble base pairs. So these thin vertical lines represent base pairs. And so editing has been completed under the direction of guide RNA number 1. There's a enzyme that will remove that bound guide RNA, and this allows guide RNA number 2 to come in and bind to this newly edited region because it has an anchor sequence that complements the red area. And it could not bind to the mRNA until that editing had gotten to this point. So in this fashion, this controls the order in which these guide RNAs carry out the editing until we have the entire mRNA edited. So my lab was interested in trying to study the structure of these different components, different functional domains. The central part's interesting because we have all kinds of mismatches between the mRNA and the guide RNA that control the editing. There's also a third domain I haven't mentioned yet, the poly-U tail that's added after transcription. This tail is added without a template. A polymerase will add on average 15 uridylates, and with up to 25 at the most, and maybe 6 at the smallest number. The mean is around 15. And these form a helix with the unedited mRNA. So this RNA in this region has yet to be edited. It's very rich in A's or G's, and as a result, we're going to wind up having a lot of GU wobble base pairs. Those are known to destabilize the RNA double helix, and we think that this unstable helix helps to temporarily stabilize the complex between the guide RNA and mRNA, but it's not too stable, so it can't be easily shifted as editing progresses. So this U poly-U tail has to migrate as editing progresses. So we went about trying to characterize what we call the U helix, as well as some of these editing site RNAs. So this is our workflow. We'll go about crystallizing RNA or protein in my lab. So in the upper left is a crystal of an RNA. It's about 0.5 of a millimeter in length and about 0.1 millimeter in width. So we'll mount that under a microscope in a cryo loop and put that in a cold stream. And then we have an X-ray generator in the lab next to my office, where we have X-rays generated on the left. We have a collimator that focuses the X-rays. And then we have a cold stream where we place the cryo loop, and we have it placed on a goniometer that rotates the crystal. So it's rotating the crystal about maybe 1 degree per every 10 minutes. And so after about 16 hours, we'll be able to collect a full data set of diffraction spots. So the image on the upper right shows is a diffraction image. This white shadow is due to the holder for the beam stop. We have a piece of lead that blocks the main X-ray beam. Otherwise, it would damage our image plate detector. This is technology that is about 25 years old. That cost hundreds of thousands of dollars at the time. But it's very useful to have available for us to screen crystals for their ability to diffract X-rays. So then we take this diffraction pattern, which essentially is a Fourier transform of the electron density in the crystal. We do an inverse Fourier transform to generate this chicken wire electron density map that's contoured at the 1 sigma level above the mean electron density in the unit cell. And here we have this chunk of electron density, which you can see is not very high resolution. This is about a 2 angstrom resolution map. We're able to model a drug molecule, and this one binds to to RET kinase, a enzyme that gets activated at the start of tumor growth. And we've been working in collaboration with another lab on trying to study why certain mutants of this kinase are able to bind some drugs but not others. And so we're in this lower right, we're making comparisons of 2 structures to sort that out. So how do we get these crystals? So we go about growing them by vapor diffusion. We will set up a crystallization drop that has in a 1-to-1 ratio our protein sample and reservoir solution. That reservoir solution has infinite number of possible components in terms of various salts, alcohols, polyethylene glycol that can cause the protein or RNA to lose, have a reduction in solubility that leads to it going into the crystalline state. So this diagram is known as a crystallization diagram. It's not a phase diagram, but it's somewhat similar. We have the protein concentration on the y-axis and precipitant concentration on the x-axis. The blue area corresponds to getting nothing. You're below this black line that represents a solubility curve. And so the protein is not concentrated enough yet, and the precipitant's not high enough to get into the green zone where we have supersaturation of the solution. So the second outcome is yet another clear drop because the supersaturation is not high enough to get nucleation to occur. The third outcome could be just immediate precipitation of the protein because the protein concentration was too high or precipitant concentration was too high. That's indicated by this red region. Or the fourth outcome that's quite common is to get a shower of microcrystals that are not useful for diffraction. The ideal situations indicated with 5, where we set up the drop initially at this point, and then the drop concentrates, and we cross this dashed line into the spontaneous nucleation region. We get 1 nuclei, and then all the protein in the drop can be used to support crystal growth. And so the crystal grows larger as the protein concentration solution goes down until we drop down to the solubility curve. And then further crystal growth halts. Of course, we can push it forward again by adding more precipitate to the reservoir. It's something that we do routinely to enhance the size of our crystals. So we do this sometimes with robots, sometimes with just manually. And in the lower left are a couple crystallization trays. The old-fashioned kind of tray uses a cover slip that's held in place with a vacuum grease. And you put the drop on the cover slip, flip it over the reservoir. So that limits the size of the drop that you can work with. So it's usually under 10 microliters. The alternative are these screw cap cover slips that allow you to easily go back in to the system. You could open up the reservoir, add more precipitant, for example. This image shows a crystallization robot that we have in our lab and that we use for screening, because often we don't know what the initial crystallization conditions should be. We have to find them. So to discover them, we vary all these variables. And so we have to search through a n-dimensional space, and often using very biased approaches, biased by prior success. And the robots are very helpful for the screening process. We eventually find a lead indicated by that star. We then zoom in to that, around that region in crystallization space, and set up further experiments to more systematically sample around the lead to find conditions that will lead to larger crystals that are of use for diffraction studies. So what are crystals? I think everybody here knows this already, but just as a reminder, a crystal can be divided up into unit cells. This is the basic small subdivision of the crystal that you can make, that you can use to build the crystal back up by simple translations in the X, Y, and Z directions. So here we have an array that's 3 by 3 in terms of these elongated unit cells. We have inside the unit cell, we have actually, we can have rotational symmetry, of course, mirror symmetry for non-biological molecules. And then we also have screw symmetry, where we have a combination of rotations and translations. So these are actually, these molecules that are 4 different colors are actually a RNA hairpin, and they have, they're packing side by side, and they're related to each other by a screw operation. So knowing that we have the same, we have this array, 3-dimensional array of molecules where we have the same atom in the same relative position in each of these unit cells. This supports X-ray diffraction. So when we have incoming X-ray beam that hits a set of same set of atoms, that in this case the distance could be the length of a unit cell edge. And if the incoming X-ray has an angle of theta, and the difference in the path length corresponds to the wavelength, or at least one integer value of it, one integer multiple of it, we can satisfy the conditions for diffraction to occur. This will lead to a diffraction spot. So every set of scattering planes will give one diffraction spot. There are a large number of possible reflecting planes, each associated with a different spot. So these are given indices called Miller indices, HKL. So we have, for example, a unit cell shown on the left with A edge, a B edge, and C edge. This corresponds, if the unit cell has 90-degree angles, this would correspond to the X, Y, Z coordinates. And we have the set of planes that has a Miller index of 0 to 0. This is because the B edge is bisected, it's cut twice, and but it's not cutting the A edge or the C edge. So we could actually have a set of Miller planes that go through the B edge 100 times. If the length of the B edge is 100 angstroms, then those planes will be separated by 1 angstrom. So these Miller indices can be actually quite large in number when you're dealing with high resolution data from large unit cells. So the main idea here is each reflection that you're going to see on these diffraction images corresponds to one set of Miller planes. So every diffraction spot is, we process the data, we count up the measured intensity, take the square root of it to get the structure factor, and each structure factor is recorded in a file, flat file that has H, K, and L indices, followed by the value of the structure factor, and then the fifth column contains will contain a sigma on the F, and then there may be a sixth column that contains a phase angle that's associated with this structure factor. So this is we just simply sum over all the structure factors by their HKL indices, and we have plugged in here in the kernel of this expression the position in real space in terms of X, Y, Z, and so we can determine the number of electrons per cubic angstroms at any given point within the unit cell by using this equation. So this diffraction pattern has, say, this dashed line represents the H-axis in reciprocal space, and then we have, say, the K-axis going in the vertical direction. And at the center, we have a spot that would correspond to the direct beam, which has a Miller index of 0.000. We can't measure that spot because it's coincident with the direct beam. So as this figure just shows the effect of collecting more data. In this case, maybe the crystal goes only 3 angstroms in terms of how far the spots go out. And we get a corresponding low resolution view of the molecule. You can see that we can hardly make out the bump associated with this backbone carbonyl. And this is kind of a high resolution cryo-EM map in comparison. We call this low resolution in crystallography. This is a medium resolution map. This is would correspond to the average structure in the protein data bank. And you can see that clearly the backbone carbonyls represented by the red for the oxygen are clearly discernible. And then we have thousands of protein structures that diffract to atomic resolution. 1.2 angstroms or better, where you can resolve the individual atoms in electron density. When I talk about resolution, it's very different from the term resolution that's used in microscopy. Our resolution is lower when we're talking about crystallographic resolution limit. So a 1.2 angstrom crystallographic resolution would correspond to optical resolution of probably 1 angstrom or 0.9 angstroms in terms of the minimal distance between 2 objects that you can resolve. So to collect the data to high resolution and also to be able to collect it faster with more modern and expensive instruments, we take advantage of national laboratories. There are 5 in the United States, 4 are supported by DOE, and the one at Cornell is supported by NSF and industry. So I happen to be this, the one that Christian mentioned. SSRL is one that we use frequently. So we have to prepare our crystals in the lab in Oklahoma. We put them in a dry shipper, and after cryoprotecting them, and we send them off to California. And then we log in to our computer at home in Oklahoma City and run a $50 million instrument through the World Wide Web and collect data for several days. This is a crystal that's clean of any frost, very unusual. You're looking at 2 views of it. It's a plate-shaped crystal that we had to reorient by bending a piece of wire that the loop is glued to. This gets the crystal in an orientation that will allow us to collect complete data to atomic resolution. The white boxes on the right correspond to the X-ray beam. We can control the shape and size of the beam at the beam line by using the beamline control software. So this is part of the software that we use. It was developed at SSRL. They developed this remote data collection technology about 20 years ago, and is now used around the world. So on the right, we have our Pilatus detector. This provides real-time readout, single photon counting, and so we can collect images at a rate of a fraction of a second, and we can collect a couple thousand images in about 10 minutes versus having to spend 16 hours in the lab next door collecting a data set that doesn't, where we can't measure the data to as high resolution. Then this yellow object is the goniometer. They have this massive motor that can control the rotation of the crystal with high precision. And then on the left is the collimator out of which comes the X-ray. And this is the beam stop. So we're able to control the geometry of the experiment remotely. This shows this is a view of an image that was collected recently. The image that is queued up and being recorded is in red, and we can plan the experiment by setting up the parameters here. So now, what have we done with some of this data? I'll talk more about the motivation for studying these poly-U's bound to the mRNA. We had, some prior knowledge existed. We had known since the 60s that poly-U's form random coils in solution, whereas poly-G's, poly-C's, and poly-A's form single-stranded helices. So these polymers can be synthesized at up to several thousand bases in length. And more recently, Lois Polak at Cornell has done biophysical studies comparing poly-U to poly-T from DNA. And she was able to, with more modern methods, establish that yes, poly-U forms a random coil that has properties that are stiffer than that of poly-T in comparison. This single strand is from actual crystal structure that has 16 U's, and so we just took this out of a structure where we had 2 strands, and so we were actually able to crystallize this in spite of this tendency of the U's not to stack on each other. So these black lines correspond to U's that are not stacking, and so that the polynucleotide chain is forming a random coil. It's thought that one hypothesis is that there are unfavorable dipole-dipole interactions between adjacent U's, which prevents them from stacking in solution. Then the other prior that we had was that GU wobble base pairs cause the double helix to be less stable. And one way of trying to understand this is to compare the structures of a GU base pair to a GC base pair. So this is the familiar GC Watson-Crick base pair, 3 hydrogen bonds represented by dashed lines. You take that base pair, flip it over, and superimpose it, you can see sort of a pseudo twofold rotation axis that relates one side of this image to the other. And but we don't have that when we do the same kind of comparison with a GU wobble base pair, because we have the U rotated out towards the major groove. So this is a view down the double helix. We're looking at just one base pair in isolation. This side of the base pair is called the major groove, and this other side is called the minor groove. So to be able to capture this elusive poly-U tail in a crystal structure, we use the trick of fusing 2 of these poly-U polypurines together head to head. And by doing so, we introduce a molecular dyad represented by this elongated oval representing a twofold rotation axis coming out of the page. So this RNA strand on the left is related to the one on the right by twofold rotation. And this means that we can assemble this fused RNA from a single RNA species. So it makes it much easier to set up the experiment. And it also gets away from the problem that has occurred in the past with heteroduplex RNAs, where the two strands are of different identity. The ends of the duplex can appear to be identical to the crystal lattice. So your heteroduplex can crystallize like this or like this, and you'll see a combination in the crystal structure. And this, unless you have super high resolution data, this could be very difficult to refine. We collected diffraction data on this poly-U RNA, which we call the U helix, and we use synchrotron radiation for that. SSRL started out as a high physics facility in the early 60s. It was used to characterize several subatomic particles, led to several Nobel Prizes in physics. Then in 1973, it was repurposed from being a particle collider to just circulating electrons continuously near the velocity of light. And so the electrons go around this dashed circle. We have a booster ring over here that's used to pump this main ring with electrons. And as they go through a system of magnets, they're accelerated. And as they're being accelerated, they give off radiation. And since charged particles experiencing angular momentum give off radiation, they give off a wide range of radiation from UV to hard X-rays. And we're able to select out specific wavelengths for use in our experiments. We crystallized a second RNA as a control, where we replaced all the GU wobble base pairs with GC base pairs, regular Watson-Crick base pairs. And we collected data in-house that were of similar quality to what we collected at the synchrotron. So we could make a fair comparison between these two. This is a controlled experiment, hardly ever done in biological crystallography. So we compare we show the electron density map with a single strand that's in the asymmetric unit for the U helix and for the Watson-Crick helix. And you see that the model is completely covered by electron density. We don't have, in other structures, there's been, the ends of these duplexes have been disordered. We didn't have that problem. You compare the two structures on the right, as we have after generating the second strand by applying the crystallographic symmetry operator. It looks like the structures are pretty similar, but if you compare the root mean square deviation between like positions, actually there's a lot of differences. And these differences are greater than the coordinate uncertainty. So the coordinate uncertainty is about 0.1 angstrom for each structure. So the combined uncertainty would be about 0.2. And you see that's quite less than the smallest difference. And these differences aren't just due to the presence of the GU wobble base pairs. If you remove them from the analysis, as represented by this dashed line, we still have large differences present. So these differences are also reflected by looking at the geometry of the double helix. You can measure the angle between adjacent base pairs, known as a helical twist. And the expected value is about 33 degrees, because we've got 11 base pairs per helical turn in A-form RNA. Our control sequence has about that, and except that there's one site where we have a magnesium binding to the backbone and distorting the crystal structure. But in the case of the U helix, represented by the black line, we have variation in the helical twist, and this variation, for example, is pretty consistent, so it's base sequence dependent. AG base step will have low helical twist values, and GA base steps have high helical twist values. So the presence of these GU wobbles is having a big impact on the structure. And then in meantime, we went about crystallizing a bunch of other lengths of RNA. with these poly U stretches, and we hit upon a length that gives us a new view of the A prime form of RNA. So this is pretty exciting because it's shown in the middle. This is the actual crystal structure. We have a model of the A form, same sequence, same length on the left, and the corresponding helical parameters at the bottom. And then same length in the B form. RNA's never been found in the B form. This is the primary form of DNA that's found in cells. But what's striking is that the width of the major groove is pretty similar in the A prime form as in the B form. So the main paradigm and in terms of how proteins interact with RNA is that they bind to the backbone. They recognize the overall shape of the RNA. And whereas on the other hand, with the binding of proteins to DNA, they often read out the base sequence through hydrogen bonding through interactions with the bases in the major groove. The fact that the A prime form has a wide major groove means that this is also a possibility for protein recognition of RNA sequences. So, so we found in our 3-tumor RNA that I showed earlier a patch of sequence. We had a number of GU wobble base pairs in a row that had features of this A prime form where the major groove was wider. So we went about conducting, and this is represented in this box. We went about docking 250,000 compounds against that site to try to find compounds that will bind with high affinity in the major groove. This is a known RNA binder, and that was docked into the major groove of our crystal structure by my graduate student. And he went about getting ranking all these 250,000 compounds by their binding energy. And so the top 5,000 compounds are going to be very similar in reality in terms of their differences in energy are within the air of they overlap in terms of the air. So what he did instead was cluster those 5,000 compounds into similar chemical groups, and then he took representatives from those groups and he did MD simulations on those to see which compounds actually remained bound and which ones wobbled off. So he found 15 compounds that remained bound, and then he went about characterizing them experimentally, and he found these 5 compounds that on benchtop experiments actually remain bound to the RNA. So we're doing follow-up studies on this. In terms of frontier methods in the use of X-ray photons and structural studies in general, you've probably heard about cryo-EM and the cryo-EM resolution revolution with improvement of detectors. This is being applied to large protein complexes. And then there's cryo-ET, which allows us to get views of complexes in cells to a resolution of about 20 angstroms. So these are both developing areas and are very exciting. And another development is with small angle X-ray scattering. This technique does not require crystals nor cryogrids. You can study the shape of molecules in solution. You can get the overall globular shape and information about its flexibility by studying small angle scattering. Another upcoming method is microelectron diffraction. This allows you to use nanocrystals that are too small for diffraction work or for work with X-rays, but if they're only a few hundred angstroms in thickness, you can use electrons and collect diffraction data with electrons to get high resolution data. This is actually a very common method now for studying the structures of small molecules, and it's being applied to biological molecules. And then there's the free electron laser, which is 1 million times more intense than synchrotron radiation, which in turn is 10,000 times more intense than the radiation I can generate next door, where you dribble crystals into a X-ray laser, and the crystals explode upon being hit by the laser, but before they explode, they give off a diffraction pattern. So you have to merge together tens of thousands of diffraction patterns from tens of thousands of crystals. But this is a way of studying crystals that or structures that can't be studied by other methods. Now, another frontier method that is related is serial crystallography using synchrotron radiation, where you shoot a crystal at room temperature with synchrotron radiation. Maybe you can collect a few images from one crystal, then you go on to the next crystal. This allows you to collect data. It allows you to study structure at room temperature. Maybe there's some artifacts induced by the cryoprotection, and there have been several examples of that have come up recently. And then finally, there's neutron diffraction. Cryo techniques have been applied to neutron diffraction and are making it more accessible. This is a photo from the LCLS, from the tunnel that's used to house this laser that's 2 kilometers long, and it's used to generate the intense laser. Now I'd like to switch to talking about PyMOL and how we're trying to make it a little easier to use. So PyMOL is a molecular graphics program that's used to display the results of crystallography. So that same kinase I've been talking about we've loaded the coordinates into this program in this viewer, and we can rotate this molecule to examine it. But the power of PyMOL is that you can generate publication quality images in just a few mouse clicks. So I clicked it on this A button for action. I get a menu, I go down and select preset, and I select publication. So in a few mouse moves, I've been able to generate a figure that's of publication quality. We have these fancy alpha helices represented by these red coils with the white on the inside and so forth. So this is for very simple use for obtaining globular views of your structure. This is very useful. PyMOL is also very easy to use to generate molecular movies. But to go beyond that is a little more difficult. You need to use the PyMOL macro language. These files have the extension of PML. I don't know if this is really the meant to represent the PyMOL macro language. I haven't seen that documented anywhere, but that's what I'm calling it. And it has a very simple syntax, a subset of Python, and that most users of PyMOL are not programmers, and they can deal with these kind of commands. You can mix in Python code. At the bottom here, we have the CMD class, the command class, imported from PyMOL. At the moment, it's commented out. And then we have the set method, and and then we have these 2 arguments, valence and off. So we're turning off the display of representation of double bonds, triple bonds, and we're just showing all bonds as single bonds. So I developed a library to make it easier to use these code fragments from a text editor. So VS Code is a very popular text editor, and this little movie demonstrates using a tab trigger. So you type in the name of the code snippet, and you get some information about it in a window. If you want to commit to keeping that snippet, you then hit tab. That's why it's called a tab trigger. And then there are all these tab stops within this chunk of code. I realize some of you already know this, but I'm shocked at how many people don't. I've presented this to some very experienced programmers who don't make use of snippets. But having the ability to go to these tab stops and change a value that gets mirrored to other sites reduces the errors bugs that you can introduce in your code. And so the tab stops also indicate sites that you should consider changing when you're reusing a chunk of code. So I made 250 of these snippets in this PML language, and I made these available for 23 different text editors, because I know that nobody wants to change text editors. Once you committed to a text editor, you really want to stick with it. And so that's available on GitHub at this site, and I've got a paper associated with that. Then I took a step further, and I made this. I have a lot of students who do not want to see code, and a lot of biomedical students. And so for them, and for anyone else, actually, I made shortcuts. So for each of these chunks of code, I made a little alias. These are actually Python function names. And so you enter capital A-O, you get the effect of applying 16 lines of code to generate this ambient occlusion effect that's not available in PyMOL. And so you can ask ChatGPT to get you code to do this, and it will return a Python command to execute AO, but it will do nothing for you unless you have my file, PyMOL shortcuts.py, loaded up in your session of PyMOL. In the middle, we have a grayscale image. PyMOL has no support for grayscale. But grayscale is sometimes called upon when you're producing a publication for like a book chapter where the publisher doesn't want to publish color, they just want to publish in grayscale. I also have a command to generate just grayscale images, and can be applied to any molecular object. And then this is a different version of the ambient occlusion effect with the carbons colored black. So you can imagine Linus Pauling standing next to something like this kind of figure. Okay, then beyond that, I have recognized that you can generate these images inside of Jupyter notebooks, and so I've developed libraries of this code as Python code that can be run in Jupyter notebooks. So the Jupyter notebooks support several different snippet systems, and provide support for a couple of those. This one system has a set of cascading pull-down menus. In this case, we're going to select under nucleic acids this DSSR block. It's going to generate a certain kind of cartoon where the bases are colored differently. This is a corresponding code in our Jupyter notebook cell. Run that cell, and we get a PNG file written to our home directory. We then load that up with some code that calls IPython, and the image gets inserted in our notebook. It's stored in our notebook. We can send the notebook to a colleague without having to attach an additional external file. You can use this to try out alternative parameters in the code, and then make a gallery of images, compare the images to decide on which one you want to include in the publication. You can also use this approach to provide support for reproducible generation of images of molecules included in papers. So these notebooks could be included in the supplemental materials of manuscripts. Our tool chain, computational tool chain for generating crystal structures is very complicated, involves about a dozen different software packages, involves massive data files. I don't really see reproducible research actually being feasible for much of structural biology, but this one aspect of generating least reproducible molecular images can be possible. So in summary, I've showed you how RNA structure is being used in our lab to do drug discovery. And I've mentioned how we're using synchrotron radiation and how it allows us to collect diffraction data much more rapidly. We can do a year's worth of work in just a couple of days. And then I've talked about this PyMOL shortcuts.py script. If you're a PyMOL user, I highly recommend it. It'll make you into a power user. And then two other libraries of code fragments, one for text editors and this PyMOL PySnips library for use in Jupyter notebooks. I also have available a library for use in CoLab notebooks. I just want to put in a plug for my institution. We have graduate programs in biomedical research. So we have 5 basic science departments at College of Medicine, and then we have a pharmaceutical sciences department in our College of Pharmacy. All of our graduate students go through the same courses in the fall semester, and then they take specialized courses in the spring semester. And our strengths are in cancer, diet, and diabetes principally, and these are some other areas. We have for example, eye institute, Dean McGee Eye Institute. And I'd like to acknowledge people in my lab who did the work over the years. My collaborator on the trypanosome work is at Texas A&M. And let's see, these are the beamline scientists that have helped me out at SSRL. I have been supported by, as mentioned before, by the NIH, NSF, and by the Oklahoma Cobra in structural biology. This is a grant that's in its 12th year, and it's a large NIH grant that has been used to help build up the structural biology program in Oklahoma. I'll be happy to take any questions.
Any questions from the audience?
Did I lose anyone?
Nice complementary topic, because I think a lot of the folks in the audience, although imaging science, they probably don't know enough about the area of application of imaging in biopharmaceuticals and drug design. So I think it was a nice exposure to potential avenues in the future with skill-based and imaging profile. Yeah. So I have a question for you. So you mentioned the drug design. So what's the next step after this is done? What's sort of the next step in the process of drug design?
So we've identified by experimentally in vitro in test tubes which compounds bind to the RNA. And then the next step is to figure out which of these compounds actually, do a number on the trypanosomes. And so it would be cellular assays to figure out how they impact the trypanosomes. Do they kill them? And then the step beyond that would be to take those that are effective and then go back and look at how they're binding the RNA by co-crystallizing them with the RNA, getting the crystal structures of the complexes. And then maybe modifying the the drug compound further so they can bind more tightly or and so on. So you start that cycle of optimization of the drug.
But before this was enabled through computational biology, how was this purely done before? What were the ways to do the different things without it? You didn't have the computational mechanics when they were not where they are today.
Wow. Very, very hard to do. So we have to go back to 1960, and people would just try by trial and error, pull things off the shelf and try thousands of compounds, do a massive amount of work. And so they were sometimes effective in doing that. And but they had knowledge, at least of the structure of the drug compounds through crystallography. And they could use that to make inferences about what's going on. And we now have the structures of the RNA that we didn't have before.
So I guess if I were to think about it, so what kind of computational infrastructure do you need for this?
I can do much of the work on my laptop. So some of the work in terms of trying to determine the structures of the RNA, say with using cutting edge methods. So I showed you this setup where I was going to collect, I didn't say it, but like 16 sets of data. I was trying to boost the strength of a extremely weak anomalous scattering from the phosphorus atoms in the RNA. I was using a very long wavelength X-ray radiation for that purpose. And to do the phasing experiments, generally only maybe 1 out of 10,000 or 1 out of 100,000 experiments actually lead to a useful set of phases that can generate electron density map. And I use the servers that at SSRL to do that work. If I had to do it locally, if they didn't let me do that, I would use our supercomputer where we have like something like 17,000 GPUs available. I've used up to maybe 1,900 CPUs, CPUs at a time. We have very few GPUs available, just a couple dozen. And so that is our weakness right now, is not enough GPUs. And for some of the drug design software, for most of it, the docking that we did, we just used CPUs. And we had to use the supercomputer over a course of months because of limitations. We could only run, on average, about 300 CPUs at a time. And each docking experiment takes about 4 or 5 minutes. So we sample 99 different orientations of the drug compound in the binding site. And so it's just a lot of wrangling of the files and managing the files and running the jobs that actually takes, you can automate it only so far, but you have to do some manual intervention. We're limited to 48-hour runs. We can't run longer than that. So in terms of the jobs that we submit to the queue, so we can only, load up so many thousands of drugs to fire off at a time. To get around some of these problems, I've gotten time at the San Diego supercomputer.
So when the pharma companies usually start developing, what stage in the process do you, could you actually get one over?
Yeah, see how far off. Probably not too terribly far off from that. So we probably will wind up using these compounds to, verify the inhibition of RNA editing. We'll use it as a tool to study RNA editing. And I think if it's useful as a tool, then that's going to garner further interest. Treating trypanosomiasis isn't on the top of the list for the NIH in terms of funding because there's only so many people that are infected with these parasites. So they do threaten the lives of people in the mid-latitudes. About 600 million people live in areas where these are a problem. You've heard of African sleeping sickness caused by Trypanosoma brucei, and then Trypanosoma cruzi causes American trypanosomiasis. We have 250,000 people infected with those parasites as a result of immigration, and there's a need for better drugs to treat them, but they're not rich people, so it will be probably the federal government that winds up funding most of this work to the point where it will become attractive for pharma to pick it up.
Well, thanks so much. Any other? Oh, go ahead.
Yeah. So, if I understood correctly, as part of your research,
I think there's a lot of applications that could be made of AI in our work. So this, like in the data processing, somebody recently started developing a neural network to help with the processing of this weak diffraction data and has success doing that. In our lab, I think that we could be using neural networks in experimental design, where we currently use linear least squares. You can substitute simple neural networks. Let's see, obviously there's AlphaFold, and that has an impact on us because that provides us, we can use that to get starting models for our work when working on new structures. I think there are many other opportunities that we haven't realized yet.
For those of you interested in machine learning applications, one of the pharma companies that hold you, they're paying more than Apple, more than Genentech, more than NVIDIA. Any other questions or comments before we end up this? Okay, well, thank you once again for your.
Welcome.